Oh, hey guys, didn't see you there. Welcome back to Bite-Sized Biology, where we talk about biological chemistry one bite at a time. Today, we'll be talking about carbohydrates, specifically carbohydrate color reactions and derivatives. But before we get to the good part, we need to establish some learning outcomes that we need to achieve by the end of the session. So first, you need to describe the different color reactions of carbohydrates and their associated chemical reactions. Then you need to demonstrate that understanding through a virtual laboratory simulation. Next, you need to both identify and differentiate the different carbohydrate derivatives. And lastly, you need to appreciate the significance of both the color reactions and the derivatives in the medical field, then formulate a short discussion on the applications of their chemical reactions. Carbohydrate color reactions are actually used in various fields of work, like the food industry, bioscience, and even the medical field. They help to identify carbohydrates in different samples and even quantify how much of it is in them. First up, we have the Molisch test. This is commonly known as the general test for carbohydrates. This works by means of a condensation reaction between the alpha-naphthol in the reagent and the furfural derivative. Now you may be wondering, what even is a furfural and an alpha-naphthol, and how are they not linked to one? Well, a furfural is formed from the dehydration of pentoses under acidic conditions, whereas alpha-naphthol is derived from naphthalene and are organic compounds. There are also phenolic compounds where the hydroxyl group is attached to the first carbon atom of the naphthalene ring system. And when the alpha-naphthol is combined with sulfuric acid, it reacts with the carbohydrates to form a violet-colored ring. See? Moving on, we have the Moore's test. It's a pretty funny name, isn't it? But what is it really? Well, it's actually pretty simple. The Moore's test is commonly known as the test for reducing sugars. Well, what are reducing sugars? Well, we have glucose, fructose, and the likes of which I'm sure many of us are familiar with. And it's based on the liberation of these aldehydes to polymerize and form a resinous substance that we all know and love as caramel. But how does it form? Well, when a solution with a reducing sugar is heated with an alkali, let's say NaOH, then the solution turns from yellow to orange to eventually a dark brown, where the odor of caramel is prominent. Our next two tests for reducing sugars are the Felling's test and the Benedict's test. Now, what makes these two tests different from the Moore's test? Well, both of these tests are an indicative of whether or not a solution with a reducing sugar is able to reduce the copper 2 ions from the copper sulfate in the reagent to the copper 1 ions of the cuprous oxide, which precipitates out of both the Felling's and Benedict solutions as a red precipitate. In any case, you can tell the Benedict's test is different from the Felling's test because the Benedict's test is commonly used to detect glucose in urine, which should actually help to detect diabetes. Next on our list is the Barfoed's test, which is, yet again, another test for reducing sugars. This time around, the reagent we're using is the copper 2 acetate and acetic acid. This is usually used to distinguish monosaccharides from disaccharides, based on whether or not the sugar can reduce the copper ions in the copper 2 acetate to form the cuprous oxide precipitate. However, if boiled for long enough, the disaccharides can actually hydrolyze into its monosaccharide components, which would then react with the copper acetate to form the cuprous oxide. Next up, we have the Solowanov's test, also known as the resorcinol HCl test, named after the reagents it uses because apparently we as a society aren't capable of coming up with anything better. This test is primarily used to detect ketoses and differentiate them from aldoses. The hydroxyl methyl furfural in the ketose reacts with the resorcinol in the Solowanov's reagent to produce a red color in the solution. These next two tests are used to differentiate pentoses from hexoses. Pentoses from the word penta, meaning 5, and hexoses from the word hexa, meaning 6. With that in mind, we know that pentoses have only 5 carbon atoms, while hexoses have 6. They work similarly, but they have different reagents as well as different indicating results. On one hand, we have the Tauber's benzidine test, which, as the name suggests, uses a benzidine solution to react with the pentoses to produce a violet-colored solution. This is actually a very sensitive test and can detect up to even just 0.01 milligram of pentose either in its free or combined form. The second test for pentoses is the Bios orsinol HCl test. The Bios reagent, which is made of orsinol and hydrochloric acid, is used to react with the pentoses and produce a green or blue solution. Both of these tests, as they work in acidic conditions, dehydrate the pentose into a furfural, which is what reacts with the two reagents. These tests can be used to detect pentoses in nucleic acids because of the presence of ribose in RNA and deoxyribose, which is present in DNA, which we'll be talking about later. Finally, we move on to our last test, starch. The iodine test is used to test substances for the presence of starch, or perhaps how well starch is being digested based on what color it produces upon the addition of the iodine solution. As it starts off, when you add iodine to starch, it gives off a blue-black color in the solution. As it breaks down, each stage indicates a different color. In order of its stages from the least to most broken down, amylose gives off a violet color, erythrodection gives off a red color, acrodection gives off yellow, and maltose or glucose do not produce a color change. If anything, it only makes the iodine a slightly lighter color. And that's it for the color reactions. If you want to try these out for yourself, click on the link over here. Alright, it's time to check your understanding. First up, what is the name of the general test for carbohydrates? That's right, it's the Molisch test. Next, this is a test for reducing sugars that is also used to detect diabetes. You got it, it's the Benedict's test. Lastly, in the Moore's test, the result produced is a resinous substance. What is that resinous substance? Of course, how could you forget it? It's caramel. Moving to the next topic of today's video, carbohydrate derivatives. Carbohydrate derivatives are sugar molecules that have been modified with substituents other than hydroxyl groups. First, we have phosphoric acid esters of monosaccharides, also known as sugar phosphates. From the name itself, it's basically a sugar molecule with a phosphate group attached. These phosphoric esters of monosaccharides occur as intermediates or precursors of metabolites, especially during carbohydrate metabolism. A good example of this is in the process called glycolysis, or the breakdown of glucose. The first reaction involved in glycolysis involves the enzyme hexokinase, which catalyzes phosphorylation, or addition of a phosphate group to a glucose, which results in the sugar phosphate glucose 6-phosphate. Next up, we've got amino sugars. Just like sugar phosphates, you can tell from the name what makes it different from typical carbohydrates. Amino sugars are obtained by replacing a hydroxyl group of a monosaccharide by an amino group. In naming amino sugars, the suffix amine is usually attached to the names of monosaccharides. An example of this would be glucosamine, where the hydroxyl group of the second carbon of glucose is replaced with an amino group. Glucosamine is the product of the hydrolysis of chitin, the major polysaccharide found in crustaceans. Another example is galactosamine, where the second hydroxyl group of the second carbon of galactose is replaced with an amino group. Galactosamine, on the other hand, is found in the polysaccharide of cartilage. Some other amino sugars include manosamine and N-acetyl-D-glucosamine. As you can see, the latter looks a little different than the previously mentioned amino sugars. N-acetyl-D-glucosamine not only has an amino group attached to the second carbon of glucose, but it also has an acetyl group, or the COCH3 group. Now onto our third carbohydrate derivative. We have sugar acids. Sugar acids are produced by the oxidation of specific carbons in a sugar molecule. Specifically, this can occur at the aldehydic carbon, the hydroxyl carbon, or both. A notable example of a sugar acid is vitamin C, also known as ascorbic acid, which is synthesized from glucose. Last but not the least, we have deoxy sugars, which we mentioned earlier. This involves sugars that have had a hydroxyl group replaced with a hydrogen atom. An easier way to visualize it is in taking the name itself, deoxy, meaning removing an oxygen atom from the hydroxyl group, so we're left with a single hydrogen atom. A good example of this, I'm sure you're all familiar with it, is deoxyribose or 2-deoxyribose. This sugar acid is most notable for its presence in DNA or deoxyribonucleic acid. Looking at its structure, deoxyribose is derived from the pentose sugar ribose by the loss of a hydroxyl group and replacement with a single hydrogen atom. Alright, let's see if you really understood what we just discussed. First up, what is the major polysaccharide of the shells of insects and crustaceans? That's right, it's chitin. Next, how would you name a galactose sugar that had an amino group added to it? You got it again, it's galactosamine. Last but not the least, what kind of atom is being removed in deoxy sugars? Yep, it's oxygen. So whether you're doing experiments in your chemistry class, testing urine in the laboratory, or just sharing a fun fact to your friends, you'll know exactly what you're talking about. But before we end, let's review our learning outcomes. In our session, we discussed several color reactions, such as the Molisch test, the Benedict's test, and Barfoed's test. We covered the chemical processes behind these reactions, explaining how they help to identify the presence of different types of carbohydrates. So, yes. We went through various carbohydrate derivatives, including monosaccharides, disaccharides, and polysaccharides, as well as their respective structures and functions. So, that's another one checked off. For the third one, we offered a virtual laboratory simulation where you could see the practical application of the color reactions we discussed. So, that's another. For the fourth, we also compared and contrasted the different carbohydrate derivatives, highlighting their unique features and differences. Ding, 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 another one. For the fifth one, we explored how the reactions and derivatives of carbohydrates are essential in the medical field, from diagnosing diseases such as diabetes to understanding metabolic processes. So that's another one. And lastly, we have our sixth learning outcome. We had a short discussion about the practical applications of these chemical principles in the medical field. We talked about real-world examples and their implications, fulfilling our final learning outcome. Looks like we covered all our learning outcomes for this session. So tune in for a bite for next week's episode, where we continue talking about another serving of carbohydrates. Don't forget to leave a like, comment, and subscribe. Thank you for watching.